Siresh garu? Hello.
Hello. Good morning, sir.
Good morning, madam. Ela unnaru? Bagunnada?
Bagunnanu, sir. Meer ela unnaru?
Fine, madam. Thank you.
Thank you, sir.
How is the Nitte College?
Yes, sir. Nice. Going good.
Recently, I saw your achievement in Tirupati, I think. One of the presentation. Tirupati or Raipur,
Yes, sir.
already you have presented one presentation at Raipur. Anantapur or
No,
Tirupati?
Raipur I have got one award, sir. Best thesis award I have got.
Exactly.
Yeah.
Yes,
Yes, sir.
He sent the details to me, that is Raipur principal.
Oh, okay, sir.
Okay, nice.
You
Congratulations,
are
madam.
like, you have full big network, whole world, whatever the updates are happening, you will be knowing.
It is nothing like
Nice.
that, madam. Just we are maintaining guarded relationship to all. Our
Yes,
pharmacy
sir.
is a very small family.
Family, yeah. Yes, sir.
Chhattisgarh, is it audible and is it visible? Everything is okay?
Okay, sir.
Okay. Okay. Madam, shall we start?
Yes, sir, we can start.
Okay.
Good morning, everyone. Welcome to the second day of 1 week International Online Faculty Development Program. I hope day 1 section was insightful and engaging. Today, we continue our journey of enhancing our skills and knowledge, focusing on the future of pharma, innovations, and applications. Let us make this day just as valuable as first one. Now, I request our principal, sir, to speak few words about today's FDP. Sir.
Eminent speaker of the today's session, esteemed teacher, Dr. MNVL Suresh Garu, Associate Professor, Department of Pharmacognosy, Nitte College of Pharmaceutical Sciences, Bengaluru, respected faculty participants from various pharmacy colleges. A very pleasant morning to one and all. Welcome to the second day of the 1 week online international faculty development program on the future of pharma innovations and applications, organized by VV Institute of Pharmaceutical Sciences, Godlavalleru, from 3rd to 9th May 2025. On day 2, Dr. MNVL Suresh Garu will deliver a lecture on integrating herbal medicine with modern pharma, bridging tradition and technology for future therapeutics. The major objective of the today's presentation is the integration of herbal medicine with modern pharmaceuticals offers a promising approach for more holistic and effective healthcare. By merging centuries old traditional knowledge with cutting edge scientific research, we can uncover novel therapeutic compounds that enhance patient outcomes. This collaboration drives innovation, improves drug efficacy, and reduces side effects through natural alternatives. Bridging tradition and technology not only preserves cultural heritage but also accelerates the development of future therapeutics. Collaborative efforts between researchers, healthcare professionals, and traditional practitioners are essential for ensuring safety, standardization, and global acceptance. The future of medicine lies in combining nature's wisdom with the scientific precision. On behalf of VV Institute of Pharmaceutical Sciences, Gudlavalleru, I would like to express my sincere gratitude to today's resource person, Dr. Emmanuel Suresh Garu, for dedicating her valuable time for the academic benefit of our faculty members. So, thank you, one and all. Now I hand over the session to Ms. B. Satyasri Garu, Associate Professor and Head, Department of Pharmaceutical Chemistry and convener of this FDP program, to continue the program. Over to Satyasri Garu.
Thank you, sir. Thank you for your welcoming note. Now I request our faculty member, Ms. P. Bhagyareka, Assistant Professor, Department of Pharmaceutical Chemistry, to introduce speaker of today's section. Bhagyareka.
Good morning to one and all. Myself, P. Bhagyareka, Assistant Professor in the Department of Pharmaceutical Sciences, Pharmaceutical Chemistry. It's my privilege to introduce Dr. NVL Sirisha Mulukuri. Dr. NVL Sirisha Mulukuri serves as the Associate Professor and the Head of the Pharmacognosy Department at the NIT College of Pharmaceutical Sciences, Bengaluru. With the 15 years of teaching experience, she earned her PhD from NIT University in 2025. Madam was completed her master degree in pharmacy from Naipur, Calcutta, and completed her bachelor from Sri Vishnu College of Pharmacy, affiliated with the Andhra University. Her research focus on natural products and herbal formulation and nano-emulsions. She has published 23 papers in peer-reviewed journals and author for other 4 textbooks and contributes 6 book chapters with renowned publishers like Taylor and Francis elsewhere. Madam holds 3 patents. She has successfully obtained research grants from prominent bodies such as including the Rajiv Gandhi University of Health Sciences and Karnataka State Pharmacy Council. She serves as a reviewer for journals and Bentham Sciences and elsewhere, and is a member of Royal Society of Chemistry and the Association of Pharmaceutical Teachers of India, and recognized for her impactful research. She has received multiple awards and maintained an H index of 7 on Google Scholar and 6 on Scopus. By this brief introduction, I would like to invite Dr. NVL Sirisha Mulukuri, Madam. And Madam, thank you so much for accepting our invitation, Madam. And I would like to thank to our Principal Sir, Dr. A. Lakshman Rao Garu and Satyasri Madam for giving this opportunity. Thank you, Madam. And thank to all participants.
Thank
I
you,
should be
Bhagyarekha.
thankful to ALR sir first. So Lakshman Rao sir is my teacher. So from 2004 onwards, I'm seeing sir. So the same energy levels are being maintained till date. So thank you, sir. Hope we will also be like you, fully energetic
you,
and
madam.
enthusiastic towards the knowledge sharing sessions. So thank you, sir, for giving me this opportunity as well.
Welcome, madam.
So should I start the session?
You can start, madam.
Yeah. So the slides are visible, no? So with your permission, I can start the session now. Yeah. Good morning, everyone. So myself, Dr. Sirisha Mulukuri, already my introduction is done.
Sirisha,
So,
ma'am,
yeah,
please share the screen.
yeah, just a second.
Slides are not visible, madam. Please
Oh.
share it.
One second. Visible? Yeah.
Yeah,
Please
Now
put
it's
a
visible.
Yes, Madam. Visible.
You can put slideshow, madam.
Actually, it is slideshow only happening, sir, here.
And you can put this cup mark, then it may, the slide size may be increased.
at least zoom the slide, Sirisha.
Here in my screen, it is full length and it is visible. How is it now?
The same in the PowerPoint mode only.
Is it fine?
Unknown. Sirisha, ma'am, please close the slide share once and again open it. Share his entire screen.
See once.
No. Stop the sharing. Go to share.
Yeah, share screen.
Screen window or tabs.
Yeah, window.
Entire screen. Go to entire screen.
Okay, from here. What about now?
No, madam,
Okay,
it
entire
looks
screen
like...
and open the PPT.
now it is okay,
It's
madam.
okay. It's okay.
Sirisha,
Okay,
madam,
sir.
you can continue.
Yes, sir.
Okay.
Thank you, sir.
Okay,
Thank you,
continue,
sir.
Sirisha.
Yeah, okay. So good morning, everyone. Myself, Dr. Sirisha Mulpuri, going to present a session on this wonderful FDP on the topic integrating herbal medicine with modern pharma, bridging tradition and technology for future therapeutics. So initially, I'll be dealing about the herbal medicine and how can we bridge the gap. And by taking a case study, I'll be moving into the entire session. So before going to the session, I just wanted to give an image disclaimer. So whatever the images I have used here, all the images in this presentation are sourced from the internet. Like some are sourced from the internet, some are designed by us. Can you able to hear me? Hello. So can you able to hear us? Hear me?
The government கேட்க மாட்டேங்குது. போன்ல போட்டா பேட்டரி இல்ல, ரொம்ப இறங்கி. ஃப்ரீயா இருக்கீங்களா?
ஹலோ.
இல்ல மேடம், 11 மணிக்கு நான் கிளாஸ் போயிடுவேன். இந்த சாட் பாக்ஸ்ல வந்து அட்டெண்டன்ஸோட லிங்க் வரும். அதை நீங்க வந்து அமுக்கி விட்டுருங்க, அது வந்துடும். லிங்க் வந்துட்டு அப்புறம் அதை எனக்கு அனுப்புங்க. போன்ல
somebody's voice I can feel. That's what I yeah. So the images whatever displayed are being taken from the internet and all rights belong to their respective owners and no copyright infringement is intended, and images are used under fair use, Creative Commons or with permission. So the complete presentation is made only intending for educational purpose. So coming to the herbal medicine, first we should know that herbal extracts, how they are being prepared. So these herbal extracts may be from dry herbs or from the fresh herbs that are being taken by means of different type of extraction methods based on the type of components such as fat soluble, water soluble, and heat resistant and heat labile components. So apart from different methods of extraction, we have different new methods such as microwave extraction, steam distillation, enzymatic methods, supercritical fluid extraction method, and semi-bionic extraction. So semi-bionic extraction is a new term where we use different enzymes, especially the digestive enzymes with respect to the pH maintained in the digestive environment. With that pH, the enzymatic digestion will take place and extraction will give efficient isolation of the constituents. And how the herbal extracts are connected to the drug discovery. So basically drug discovery is based, or now it is having various trending methods like biochips, genomics, like where biochip is nothing but how a chip is used in a computer to operate various mathematical operations. Similar way, biochip is nothing but it is used inside our body to detect some diseases and to find that which biomarker is suitable for the identification as well as for the treatment of the disease. Biochips are being used in the drug discovery. Apart from the biochip, there are different techniques such as genomics, bioinformatics, metabolomics, and proteomics and computer-aided drug design. So how the herbal medicine is continued and connected here. What is the value of herbal medicine? Why we have to know about the herbal medicine? Because herbal medicine is having a rich source of bioactive components. We can take few examples like aspirin. So everybody, if they are having fever or body pains, like Calpol and aspirin are very well-known drugs. So this aspirin is being taken or being manufactured and isolated first and then converted into a finished product from the bark called as willow bark. And coming to the next compound, that is artemisinin. So artemisinin is used as an anti-malarial drug which is taken from the sweet wormwood. So all these drugs are mainly obtained as bioactive components initially and then converted into a final product from the herbal extracts. So this herbal medicine, it gives importance mainly with respect to the holistic approach to the health. So yeah. Okay, and then cultural and historical significance is being maintained and well established with respect to the herbal medicine. And what are the different advances in the modern pharma? So in modern pharma, if we see, apart from the invention, apart from the discovery of the drugs, rigorous clinical trials are being taking place, especially with respect to phase 1, phase 2, phase 3, phase 4. Like in the phase 4, many challenges are being faced. But after completing these 4 clinical trials, only we are getting the finished good in the market. So before that, we have to standardize the process or production of the drugs. And then advanced drug delivery systems. These are very, very important. How the delivery system, how can we minimize the dosing and how can we deliver the dose in a particular dosage forms will be known, especially in the advanced drug delivery system. And precision medicine is very, very important, where a person need to have a precision medicine for particular disease, particular medicine should be prescribed. So these are the advances taking place in modern pharma. And how can we bridge the gap between the herbal medicine and the drug discovery or the future innovation therapeutics? First thing and foremost important thing here is phytochemical analysis and standardization of the drugs. So this is the very, very important. This is the main pillar for the integration as well as for the innovation with respect to the therapeutical aspect, where if we analyze, if we standardize the particular drug in a proper manner, then only we can able to convert and we can able to integrate that particular medicine for innovation process. So how the innovation happens is by combining the herbal extracts with pharmaceutical dosage forms. So based on the solubility, based on the chemical property of that herbal extract or the molecule, then we will be designing them into suitable dosage forms. And then evidence-based research on herbal remedies. So if we see the traditional healers, they are having a lot of knowledge towards the diseases, towards the medicines. But where is the evidence? So we have to work towards the evidence-based research process, where we have to know about the remedies and how the remedy is being happening and which molecule is being responsible and why it is responsible, what are different clinical aspects and what is the dosing, what is the side effect. All these aspects will be understood by bridging the gap between the herbal medicine along with the remedies based on the evidence-based therapy. And once evidence-based therapy is validated, we have to go for regulatory harmonization. We have to take suitable approval, suitable licenses based on various authorities. Then only a finished good will come. Then only innovation process will come. So when we are going for this innovation, when we are going for bridging the gap, so we should understand case studies. So what are the different case studies? If you see, as I have discussed, is artemisinin. So artemisinin is available in the market in the form of artesunate or artemether, because artemisinin itself is not soluble and itself is not a stable molecule. So we have to convert it into a stable molecule and we have to improve its shelf life without affecting its medicinal properties. So artemisinin is used for the treatment of malaria, which is discovered from the traditional Chinese medicine. Now it is a global market. It has a global standard treatment and it has reached millions of market in the globally. And then curcumin, which is important. It's a gold. So we call it as gold in the herbal medicine or in the crude drugs. Turmeric is a golden standard, where curcumin is been isolated, which has a very potent anti-inflammatory as well as anti-cancer properties, and it is used in novel drug formulations. If you see nowadays, curcumin spray, curcumin inhalers, curcumin dosage forms are widely being used by many people, because the side effects are very less with respect to herbal medicine. So the now trend is going towards the herbal medicine. By bridging the gap between this herbal medicine and the market, we can bring out some innovations. So as I told you, we have seen some case studies. Now on one specific technology I would like to speak, which is a nano drug delivery system. It is phytoemulsion. So phyto is a plant-based molecule or plant-based extract or plant-based component which is integrated with a novel delivery system such as ML gels. So these phyto-ML gels are topical. ML gels are basically applied on the skin. So they are topical drug delivery systems that combines a gel and an ML gel and are used to deliver the herbal extracts. So the herbal extracts, if you see the maximum number of herbal extracts or the herbal compounds are the isolated compounds, many are not soluble in water. So we will have a lot of disadvantage with respect to the delivery of these drugs, especially for the topical delivery. So in order to convert them, use them for skin conditions, we can use the technique of phyto-ML gels. So they are used to treat a variety of skin conditions, including acne, psoriasis, and fungal infections. So ML gels are nothing but the infused delivery systems with the plant-derived bioactive compounds and are natural, biocompatible with minimal side effects. So they are effective for skin-related disorders and they target cancer cells very effectively. And example of one of the formulation will be discussed in the next slides. So till now, if you have any queries with respect to these slides, you can ask me. Okay, that means like I can go ahead with the next slides. So skin cancer. So as I am discussing about the ML gel formulation, so ML gels are mainly meant for skin conditions, especially the skin cancer is a main worldwide problem. It is the 17th most cancer worldwide, where abnormal growth of skin cells takes place because of various reasons, especially due to genetic mutations and UV radiations. And various types of skin cancers are melanoma and non-melanoma. If we are not treating the skin cancer in right time with right medication, it can be fatal and leads to death and leads to metastasis also. So various treatment options are available like surgery, but surgery is very expensive and leads to many comorbidities and high expected cosmetic concerns are been associated with this surgical treatment. So other than the surgical treatments, we have another option such as non-surgical treatment where using different creams and different products, we can get rid of the skin cancer, but a lot of side effects are associated with this, such as local pain, swelling, erythema, flu-like symptoms, and low clearance rates. So there is a much emergent need for the treatment of skin cancer with respect to the herbal medicine, where we are going to bridge the gap with a plant-based medicine for the treatment of skin cancer. One such molecule is andrographolide, which is obtained from Andrographis paniculata. It is a diterpenoidal lactone molecule obtained from the family of Acanthaceae, having many pharmacological aspects, especially anti-cancer, anti-diabetic, anti-inflammatory, hepatoprotective, and antiviral properties. Although it has anti-cancer properties, the molecule is not being delivered till date in the form of any skin formulations. So in order to deliver this molecule into the formulation aspect, the phyto-ML gel concept is taken. And in order to make in the form of phyto-ML gel, first you need to, any molecule, if it is insoluble, the suitable dosage form for its delivery to the topical route is phyto-ML gel concept, where initially you have to convert the molecule in the form of nanosizing. So how we have done this nanosizing of andrographolide? This is a work which we have done in our lab. So this nanosizing is done by using sonication method, where we have used sonication followed by solvent evaporation method, and where we have dispersed the andrographolide ethanolic solution into acacia solution and sonicated for 10 minutes by adding dropwise. And once the dropwise addition is over, the stirring is taking place, followed by solvent evaporation, where we will get uniform droplets, which can be confirmed by the nanosizing using SEM analysis and particle size. And after converting into nanosized andrographolide, we have to see the compatibility studies, whether any formulation when you are making with a plant-based compounds, first you need to check whether the plant-based component or the isolated molecule or the pure herbal molecule is actually compatible with the other excipients or not. So compatibility studies generally we used to do by FTIR, spectroscopy, where you can see chemical interactions between andrograph light and the excipients. So here you will be taking andrograph light as well as the other excipients like oil, polymer, physical mixture, and ML gel formulation. As I told you, ML gels are combination of emulsions and gels. Always we know that emulsions means oil in water combination. So you will have oil phase as well as water phase. So coming to the formulation aspect, so formulation of andrograph light loaded emulsion. Here we go for 3 important steps. The first one is preparation of emulsion. As I told you, it's a combination of emulsion and gel. First, we need to prepare an emulsion. So it's a mixture of aqueous phase and oil phase. So aqueous phase contains andrograph light, whereas oil phase contains oil. That oil which is suitable oil for your formulation. Here we have taken flaxseed oil and then vitamin E oil. The combination of these two will become the oil phase. So both oil phase and aqueous phase should be stirred and subjected to sonication method to get a nano ranged emulsion. Once the emulsion is prepared, next phase is preparation of the gel. So we have to make a gel phase using glycerin and water system. Here we have prepared glycerogels. So ML gels prepared with glycerin. If they are prepared with water, they are called ML gels. If they are prepared with glycerin, they are called as glycerogels. So based on the application, we can use glycerin or water. And then once the preparation of emulsions and the gel is done, we have to mix the gel and emulsion in equal ratios, like 1:1 ratio, to get the ML gel formulation in a homogenized condition. So this is the composition we have used in our lab. So where we have used different oils. Among the different oils, we found like the flaxseed oil is having very good properties. Like when we have evaluated the gels, we have seen that used the flaxseed oil formulation has given best results with respect to various evaluations and responses. And in order to know the suitable proportion of the oil component and the polymer component or the gel component, we have used a software called Design Expert. Using central composite design, we have taken like 5 level 2 factorial trials with 11 experimental runs where xanthan gum and flaxseed oil are being taken. So xanthan gum is a gelling agent and flaxseed oil is a oil phase. So dependent variables are spreadability. As I told you, various evaluation parameters we have conducted. So among them, where the parameters which we have fitted into this Design Expert software are spreadability, extrudability, viscosity, drug release for 1 hour and 24 hours. For any formulation, the drug release is the very main aspect. So if the drug release is happening, then only we can understand the formulation is working efficiently. So here, once the optimization is done, once the formulation has been prepared, we have subjected the formulation for various release kinetics to see the release pattern, whether it is following 0 order or first order. So in vitro release data has been fitted with various different kinetic models such as 0 order, first order, Higuchi and Corsi-Meyer-Papas equation. So based on the constant, so based on the regression coefficient, the pattern or the graphical representation will tell you based on the value of R square. If it is more than 1, if it is less than 1, it will tell you which release pattern is being followed for our formulations. Then followed by the release kinetics, we have already also conducted stability studies as per the ICH guidelines, where we have maintained a temperature of 40 degrees centigrade with RH of 75% for a period of 3 months. And every 15 days of intervals, for a period of 3 months, the analysis is being done with respect to spreadability, extrudability, and various other parameters to see whether the formulations are stable or not. And coming to the biocompatibility studies, once the formulations are stable, once the formulations are ready for particular release, before going to tell that our formulations are ready, we need to focus on the biocompatibility studies also. So biocompatibility studies, why I am talking about biocompatibility studies? Because our formulations we have designed for the skin cancer activity. So before going to test on the skin cancer, first we should know whether they are having any side effects with respect to healthy cells or not. So these formulations are being checked for compatibility with respect to HDF cells, human dermal fibroblast cells, where the safety profile against normal skin cells is being tested, and the final formulations notated as FIG and FFE. These are the two final formulations which we have checked to know the biocompatibility profiling, so that to know whether they are effective towards the skin, they are having any side effects, or they're okay with the healthy cells or not, is been confirmed by biocompatibility studies taking HDF cells, nothing but human dermal fibroblast cells. So once we understood the formulations are safe enough to use, then we have focused, or we have tested the formulations against cytotoxicity study. So the cytotoxicity study is being done using MTT assay, where the formulations are taken in different concentrations comparative with a standard anti-cancer drug like amphotericin and tested against this cytotoxicity study. So once we have understood that the formulations based on their IC50 value, the formulations are effective towards the skin cancer using in vitro assay method. Before going for the in vivo methods, we have also checked their permeation ability through FITC analysis. So as per the equivalent concentration based on the IC50, we have conjugated a molecule called FITC. It is a dye, fluorescein isothiocyanate dye, used for 10 microgram per ml we have taken, and we have added along with the formulation and tagged this molecule with a formulation, and this formulation is subjected to the cells or A431 cells, which are actually for the skin cancer cells, and we have incubated for half an hour, 1 hour, 2 hours, and 4 hours, and then the harvested cells are being analyzed through flow cytometry to see whether the formulation is entering inside the cells, permeating inside the cells or not. If the formulation is entering inside only, we can say that this is effective for skin cancer. Else we cannot able to say that this is effective. Based on the IC50 values, maybe it is having the cytotoxicity study, but being an ideal formulation, it has to enter inside the cells. And then before going for the ex vivo or before going for the in vivo studies, how we have conducted the compatibility studies, checking on the healthy skin cells. Same way we have conducted the skin irritation studies on the animals. So we have taken Swiss albino mice as our experimental animal model, and we have taken different blank gel formulations, and we have taken different formulations which are prepared, and we have checked the skin irritation studies for 24 hours after the sample application. Just a minute. Hope the session is audible, and is there any doubts till date? You can ask me. Yeah. So followed by skin irritation studies. Is it visible and audible?
Yes, Sirisha, you can.
Yeah. Okay. So we have done the in vivo studies, and then same way how we have checked the permeability of the formulation inside the cells, same way we have checked the permeability of the formulation inside the tissue of the animals by means of in vivo skin uptake study, where we have tagged the FITC with different formulations, and then we have checked it. So we have applied onto the skin tissues, and then we have seen the permeation of the formulations on the skin membrane of the animals. And this is confirmed by confocal microscopy, where we have understood by nanoamalgel uptake by different skin layers. In online meeting with the FDP. FDP. And the next one is characterization of nano-sized andrographite. So we have done with various uptake mechanisms. We have done with various in vitro and in vivo studies. Because the formulation should contain nano-sized andrographite, then only it will be allowed inside the tissues. So this characterization of nano-sized andrographite is done by particle size analyzer as well as SEM analysis. So we found that the particles are in nano size. And then compatibility studies also, we found that there is no interaction between the excipients as well as the andrographite, which states that the formulation is basically compatible. And then, as I told you, we have used a central composite design. We can see here various responses and various factors. So based on these factors and based on these responses, we have optimized a formulation with a suitable proportion of xanthan gum and oil. And that formulation is selected for in vitro and in vivo studies. So before going for the studies, as I told you, we have subjected it for various release studies such as first order kinetics and Higuchi kinetics. So you can see here the R square value and N value. So N value, if it is near to 0.9 or N value it is near to 1, that states that the formulation is following that same pattern. If we see the first order kinetics, 0.98, and in case of FIG it is 0.991. So our formulations are following the first order release kinetics. So slowly the drug is sustainable fashion it is releasing, which is required for cancer study. And then, as I told you, we have conducted various stability studies. When we have seen the stability studies of these formulations, every 15 days we were checking, and we found that there is no change in the color and odor of the formulations. And especially the formulations which are prepared with 6.8% flaxseed oil and 6% hip oil. These are the two important formulations we have got, which are highly stable. And always we have to remember that ML gels means the combination of emulsions and gels. So always we have a problem of phase separation and lack of stability issues. So stability issue is not existed in our technology, and also we have not seen any phase separation or no liquefaction is observed for a period of 6 months as per ICH guidelines. And we have checked pH, spreadability, extrudability, and rheological characters which are not affected. The important aspect for any topical formulations, especially for MLGs, is spreadability. They have to be spreaded very uniformly, and they have to be, see any semi-solid dosage form, the important criteria is spreadability. It should have high spreadability so that the amount of drug release, amount of the drug application towards the skin layers will be maximized. And so that we can, so that the drug release and so that the drug reaches to that particular layers leads to the therapeutic effect. Similar way, extrudability. The moment you press the tube, it has to be coming out in a uniform manner. So that is called extrudability. And rheological characters are also not affected. With viscosity parameters are also being checked. They are also ideal in both the seasons. And there was no significant difference in the drug loading and drug release studies of the freshly prepared and the stored formulations. As I told you, we have compared all these parameters with respect to the freshly prepared formulations and 6 months stored formulation between every 15 days of intervals. And if you see ex vivo permeation, so we have also done the ex vivo permeation using the pig ear skin, porcine ear skin, because porcine ear skin is having the same properties to the human properties. And we have found that the best formulations are having highest amount of the drug release. And the biocompatibility studies shows that, see here, the biocompatibility studies of these two formulations at 50 microgram per ml, 91%, 90% cell viability. That means no cells are being died. That means cytotoxicity is not there with respect to the HDF cells, human dermal fibroblast cells. And coming to the next aspect, skin irritation study. As I told you before, going further in vivo studies or animal studies, we have taken the approval from the NITE University, and we have done animal studies with respect to the skin irritation initially, where we have applied the gels, blank gels, and the pure molecule, and we have seen after 24 hours of time whether there is any skin irritation or there is any allergy or any rashes or any changes in the behavior of the animals is being observed. And totally we found that the animals are behaving normally and there is no redness or no allergy or no skin irritation takes place, which shows that the formulations are safe enough to use for skin cancer with respect to the animal models. And before that, we have also performed the permeation studies. As I told you, FITC analysis, which were confirmed through confocal microscopical studies. And the cell uptake study. See, if you see here, increasing the time, the uptake is also increasing. That means when we are subjecting the formulation and when we are incubating for a longer time, the formulation is entering inside the layers. That shows that the formulation has very good permeability. That is what observed in cell uptake studies. And this is done by confocal microscopy, where the animals are being applied. You can see here 2 hours, 4 hours, 8 hours. So after applying the formulations, every 2 hours, we have checked the skin of that particular animal and we have taken the biopsy of that animal and we have subjected it for biopsy. So we have subjected it for the imaging analysis using the confocal microscopy and image software, where you can see the darker green intensity signals are observed for higher intervals of time. That means the high intensity color is observed means high amount of drug is permeated inside the layers, because the formulation is being tagged with FITC, which is a dye, which is a thiocyanate dye. It will give fluorescence. So that is how we have detected that the formulation is entering inside the layers. Okay, so till now I have updated you about the herbal medicine, and in that one case study I have taken where in our lab only we have done that molecule which has not been delivered till date, and we have delivered successfully into for the cancer treatment, where we have seen that this is a very good ideal molecule and very good ideal formulation for the treatment of cancer study. Although we have done the study up to preclinical data or clinical aspects, so there are some challenges when you are bridging the gap between the traditional medicine and the future innovation therapeutics. So what are those challenges when you are integrating or when you are translating a plant molecule into a finished dosage forms? We will have various challenging aspects with respect to the stability concerns of the plant compounds. And also when you're talking about the innovation therapeutics, personalized medicine is very important. So how it is scopeful, how the future scope is integrated or tagged with the personalized medicine. And we need to have this newer tools. Like now we are having different artificial intelligence tools. So when we are going for these kind of integrations, we always should look into this AI and ML for drug discovery, because AI will be useful for getting the information and machine learning will help you to see the statistical data or to see how many formulations are there, how they are being scrutinized, how many things are successful. Based on all this, we can go successfully towards a new drug discovery era. And especially with respect to the plant-based or herbal-based medicine, we need to focus on sustainable sourcing and biodiversity conservation. If you're cutting the roots, if you're taking out roots of plants, tomorrow we will not have that particular plant. For example, if you see licorice root is having that particular activity with respect to sweetening property as well as with respect to the gastric protection. So all the time we are actually exploring the roots, means tomorrow for the next generations we will not have licorice at all. So what we need to do is, being in the pharmacy profession, we need to explore other than the root, the other parts of the plant is having similar efficacy or not. So by that way, we can maintain the sustainability and conservation issues. And we need to have global collaborations between the traditional healers and the scientists, because scientists will go with the scientific facts, but traditional healers will have a lot of tremendous knowledgeable insights, and they will have a lot of information about various plants. Till date, many plants need to be explored, but traditional healers are already using those plants which are
Sirisha ma'am. You are muted. Unmuted. Yes.
It is in mute.
Now it is okay.
Should we go back to the previous slides?
Yeah. Just now only you are muted.
Okay,
Cut
fine.
it. Cut it.
Okay, as I told you, we have seen the permeability of these formulations through fluorescent microscopy using confocal microscopy, where we have tagged the molecule into a formulation and we have seen the permeation. You can see here 2 hours, 4 hours, and 8 hours. So when the formulation is subjected for 8 hours, when you take the biopsy of that tissue of that particular animal and subject it for image analysis, you can see high intensity green intense signals that shows that the formulation is going inside the deeper layers, which is very much important, or which is very essential aspect with respect to the skin cancer treatments. So this is how we have merged or integrated the traditional or a herbal molecule for a finished formulations. So when we have done this integration, or when we have translated this molecule into a therapeutic innovation, why I'm telling therapeutic innovation, because with this technology we have got 2 patents. So what are the challenges and what are the future prospects with respect to this translation or integration between herbal medicine and therapeutic innovations? Are stability concerns. Whenever we are dealing with plant molecules or plant compounds, always we have to remember the stability aspects and the future scope in personalized medicine. So what is the future scope? How we can give this particular formulation or for particular dosage form for a clear disease? So clear indications and clear aspects with respect to the personalized criteria should be known. And then the new era should have a tremendous knowledge with respect to AI and ML, because based on this AI tools and ML tools, we can understand the drug discovery process, and we can avoid repetitive steps with respect to these innovations. And we should have the sustainable sourcing and biodiversity conservation. So where being a pharmacognosy field, or being a traditional dealing with the traditional medicine, we should know the sustainable aspects. How suppose example, if you see licorice always for the ulcer treatment or for sweetening property, we are exploring the properties of the rhizomes of the licorice. But all the time, if you're cutting the rhizomes, tomorrow we will not have licorice plant. So being in this pharmacy profession, being in this particular integration, all of us should explore other than roots, where other parts are also having the same property or not. We have to explore that, and we should avoid the sustainable issues so that it leads to biodiversity conservation. And then global collaboration between traditional healers and scientists is also very important, where the traditional healers are having tremendous knowledge, but they are not having the scientific facts, or they are not going with the evidence-based system. But scientists are discovering the medicines or the molecule based on the scientific facts. If the traditional healers and scientists are having global collaboration, then we can come up with a very wide variety of innovative traditional and plant-based medicines, which are having high therapeutic innovations. So as I told you, innovations. So as I discussed about a case study where we have done in our lab about the andrographlate formulations, we have got 2 patents with respect to this study. So one is with respect to its isolation using a novel method, and the second one is with respect to its formulation for the treatment of skin cancer. And also we have published the same work, different parts of the work of the entire concept into Q1 journals. So one is in the gels and one is in Saudi Pharmaceutical Journal. So finally, I would like to acknowledge God for blessings and giving me this opportunity. And the present work could not have been completed without the encouragement, assistance, and guidance provided to me by Dr. Satish Madhav, who is a scientist in the FR&D department in Vital Pharmaceuticals in Hyderabad. And I thank our principal, Dr. V. Kusumadevi, all my colleagues for their continuous support. And last, I would like to acknowledge the entire team of VVIPS and the organizing team and the principal ALR sir, Lakshman Rao sir and Satyasri madam for giving me this wonderful opportunity to have this knowledge sharing session in front of every one of you in the FDP session. Now the session is open for the questionnaire session. So feel free to ask me any questions with respect to this topic. Thank you so much.
Thank you, Sirisha, for your valuable insights about integration of herbal medicines with modern approaches, bridging tradition and technology for future therapeutics. Very nice presentation. Thank you for your sharing your valuable knowledge with us. Some questions are there in chat box. Sirisha.
Yeah. Can you see them?
Madam, any marketed MLGL are approved and what are the regulatory guidelines?
Okay, so we are seeing no Volini gel, Volini spray. We are seeing right for. Am I audible?
Yes, you are audible.
Yeah. So thing is that all of us are using this Volini spray for treatment of the pain, right? So Volini is previously manufactured in the form of ML gel. After diclofenac gel, Volini is nothing but diclofenac gel, where diclofenac is having anti-inflammatory and pain-killing property. So that's what so initially the gel is prepared, then only it has come into the market in the form of spray. And coming to the regulatory guidelines, there are no specific or very tedious guidelines for these ML gel formulations. Once the clinical trials, like preclinical to clinical data we establish for the treatment of that particular targeted disease, we can easily go ahead with this into the market. Hope I have cleared your question. Any other questions?
Androclacolide is isolated from plant or synthetic one?
No, it is isolated from plant. Andrographis paniculata. So previously, till date, it has been isolated by using column chromatography only. So in our lab, we have tested with microwave assisted extraction technique, where we have improved the yield. The yield is only less than 1%. So we have improved the yield to an extent of 4% using the microwave assisted extraction method, using less amount of solvent and less amount of time. That is how the green technology is being introduced. So that is where we have got a patent for this isolation process.
One more question. How long to maintain the stability study for MLZ?
Yeah, it is first in the initial trials, we can maintain up to a period of 1 month. But when you wanted to test it on animal studies or animals, when you have a future aspect of commercialization aspect, we have to maintain for a period of 6 months. So if you maintain 6 months based on ICH guidelines, then only we can prove that the formulation is stable. Two important aspects here. One is stability, one is efficacy. So when it is a stored long period of time, also you have to check its physicochemical properties along with the therapeutic efficacy. And all the parameters are having uniform results, then only we can bring it to a market. So this is the aspect with respect to stability studies.
One more question. What are the key formulation challenges in developing stable and effective herbal emulsions?
Yeah. So mainly since plant-based compounds, when you wanted everything natural, so definitely we will have this contamination issue and the phase separation issue, because they are combination of emulsions and gels. Always emulsions means we have two phases, like aqueous phase and oil phase. So when your molecule is either soluble or insoluble, we are having water phase in your formulation. So when your formulation is having water phase, definitely stability issues will come. So that is where you will have a key role in selecting your gelling agent. Gelling agent is nothing but your polymer. Your polymer should be a stable. So we have used in our formulation xanthan gum as well as carbopol combination. Carbopol and xanthan gum are having very good compatibility and they are very highly stable polymers. That is how they will bring up the stability to formulations. Yes.
Yeah, one more question. What are the most critical parameters to consider when selecting herbal extracts for MLGel formulations?
Critical
Parameters to consider when
okay,
selecting
first,
herbal extracts.
okay, so foremost important thing here is, first you should know in the topic, ML gels are topical formulations. So whether you are targeting the topical disease or not, that may be cancer, that may be a psoriasis, that may be a skin rash or skin allergy or any skin conditions. So if your extract is having so and so property with respect to topical study, we will select it. And second thing is whether it is having a water soluble molecule or water insoluble molecule, you have to select. So based on that property of solubility issues, then only you can choose a gelling agent, then only you can choose a phase. It can be a water phase or it can be a glycero phase. So if it is highly unstable molecule, if it is highly water, it's better to select glycerin as your phase, aqueous phase. Instead of water, you select the glycerin. If it is partially soluble, if it is not insoluble, then you can use a water phase. Hope you got my point.
One more question, Sirisha ma'am. Can nanotechnology or bioenhancers be integrated into herbal emulsions to improve therapeutic efficiency?
Yes. See, ML gels, we can do a normal ML gels, but why we are going for nano ML gels? In my study also, I have shown you nano sizing of the androgen fluid. Always we should remember that when the molecule is having the efficacy, but in larger proportion, if it is giving the efficacy, it is a need to convert into a nano molecular size. Because when you convert into nano size, what will happen? So you can decrease the dosing. You can decrease the dosing frequency. By that way, if any associated side effects are there, we can also reduce the side effects. So in my study, the nanosizing is being done by using sonication followed by solvent evaporation method. So you can use any method like precipitation method, immiscible solvent evaporation method, or you can convert into silver nanoparticles, whichever formula you wanted to use. In that way, it is better to convert that molecule into nanoparticulate size so that we can have a micromolar or nanomolar IC50 values, and we can decrease the dosing levels. Instead of using suppose 50 microgram you have to use, but if you have converted into nano sizing, you can use only 5 micrograms. By that way, you can improve your therapeutic efficiency with low doses. Yes. Any other questions?
Thank you for answering the questions of various participants. If any participant interested to interact with ma'am, you can.
Yeah, please feel free to interact with me. We have some more time. I hope the session is clear to all of you.
Thank you, Sirisha ma'am.
Thank you.
Thank you for accepting our invitation in spite of your busy schedule. Thank you once again. Very nice presentation.
It's my pleasure to have a session in front of all the participants and taking the privilege or the opportunity from VVIPS. Thank you so much. Okay.
On behalf of Institute of Pharmaceutical Sciences, we would like to present a token of appreciation certificate. Please accept it.
Okay, thank you.
Is it visible, Sirisha ma'am? Certificate.
No, it's not.
ఇప్పుడు నేనేమంటాను కదండి 10 మార్క్స్ సెకండ్ సెమిస్టర్ కి 10 మార్క్స్ వస్తుంది కాబట్టి ఏ ఇప్పుడు ఇలా వేసేసారు
Please accept our certificate. Thank you.
Yeah, pleasure in taking this. Thank you. So, Bye bye to one and all. So signing off, Sirisha Mullikuri.
Thank you. Thank you all the participants for your participation. If the attendance is not working now, I will post the new link in tomorrow's section. Thank you, one and all.
Can I leave?
Thank you, Sirisha ma'am. Okay, you can leave, ma'am.